Welcome to a new episode on the But Digestible series. Today we will talk about the cell cycle, meiosis, but digestible. If you haven't, highly suggest watching our mitosis video first to be able to understand this one. My name is Camila Pimentel and I'll be your guide today. Meiosis is a two-part process that produces four haploid daughter cells. Haploid means that they don't have chromosome pairs, they have chromosome units. They have 23 units of chromosomes at the end of the process. This is a type of cell division that enables the formation of eggs and spermatozoids. It also allows for genetic variability through a process called crossovers. Meiosis is essentially mitosis repeated twice. It begins with a cell and interphase. To review from the previous video, interphase as G1, S, G2. The G's are for growth and S is for synthesis. After G2, we have meiosis 1. Just like mitosis, it has karyokinesis or nuclear division and cytokinesis or cytoplasmatic division. G1 is where cells become committed to continuing the cell cycle or staying arrested in G0. There, proteins are built, energy reserves accumulate, building blocks are produced, and the cell has 46 chromosomes. S is where cells replicate their DNA and centrosomes. The cell now has 92 chromatids. G2 is where cells rebuild their energy reserves, build more proteins, and the cytoskeleton dismantles. Now, this picture is the cell cycle of mitosis, so it only shows one daughter cell plus the original cell, each with 46 chromosomes. Meiosis 1 is Meiosis 1's outcome is similar but not quite. Just like mitosis, meiosis is divided into prophase, prometaphase, metaphase, anaphase, telophase, and diakinesis. However, there are extra steps in prophase 1. Prophase 1 has leptotene. The duplicate sister chromatids first condense into distinct thread-like structures. Homologous chromosome pairing also occurs during leptotene, in which the homolog axes are aligned with each other, roughly 400 nanometers apart. In zygotene, homologous homolog, homologs are brought even closer together, to a distance of about 100 nanometers, in a process called synapsis. An array of protein filaments called synaptonemal complex is assembled between the homologs. Tachytene is the stage at which the synaptonemal complex is complete and their homologs are tightly linked along their entire lengths. The chromosomes become highly condensed in diplotene, and crossovers become apparent as chiasmata. Crossovers is where homologous chromosomes, attention, not sister chromatids, exchange part of their DNA. This causes enhanced genetic variability since a chromosome that originally came from the father can have a part that came from the mother and so forth. Diakinesis, here the nuclear membrane starts to break down, the nucleolus disappears, and the crossovers are done with. In prophase 1, the nucleolus disappears, centrosomes move, chromatids condense, homologous pairs pair up, and crossing over occurs. In prometaphase 1, the nuclear envelope disappears. and centrosomes move to the opposite poles of the cell. In metaphase 1, the homologous pairs align on the equatorial plane. In anaphase 1, the chromosomes are pulled towards the poles and the cell elongates. In telophase 1, chromatin relaxes, the nuclear envelope forms, and the nucleolus reappears. In diakinesis, the ring of actin filaments contracts, the cleavage furrow forms, and the cell cleaves into two halves. And then we get into meiosis too. Something important is that we don't have interface in humans in between meiosis 1 and meiosis 2. Thus, chromosomes will not replicate. Instead of sister chromatids, what's being separated here are the homologous pairs. We begin with two cells that are diploid. The chromosomes are in pairs with 46 chromosomes and end up with four cells that have 23 chromosomes. In prophase 2, chromosomes condense. In metaphase 2, the nuclear envelope disappears. In metaphase 2, they organize in the equatorial plane. In anaphase 2, they get pulled to opposite poles. In telophase 2, nuclear envelope and nucleolus reforms. In diakinesis, the cytoplasm separates. And I want to view this image again after explaining all of that. Notice how due to crossover, some chromosomes have mixed colors, or they are mixed with other chromosomes that ended up in other cells. Notice how we go from a cell exiting interface with 92 chromosomes, and we end up with just 23 in four daughter cells. I want to give a shout out to the sources. We have The Cell Cycle Principles of Control by David O. Morgan. It's a very, very good book. Unfortunately, the link right now is down. So to access it, you had to go through the Wayback Machine. I actually contacted the author and he said that they will fix the link hopefully. So that's that's it for the sources. And for the credits, we have Camila Pimentel, Rowan's editing, script, voiceover. That's me, by the way. Ivana Pimentel, script clarity. My family and dogs, moral support. And I want to thank you for being here, for supporting this channel. If you haven't yet, I want you to subscribe, to give me a like in this video, and to leave a comment. So, thank you very much and see you in the next video.